Hello, Dr. Raymer here, and today we're talking about motor control. And I just want to start by saying that we tend to think about motor and sensory control as separate entities, but of course they are anatomically and functionally linked. And we know that because sensory feedback is absolutely crucial for motor control. So that's what this diagram is showing you, is these double-sided arrows represent reciprocal connections between motor and sensory areas. And that includes reciprocal connections between primary motor and primary somatosensory cortices, between primary and association cortices of the same modality, motor and sensory, and even between secondary cortical areas for motor and sensory control. So a whole host of reciprocal connections links these areas, and it's really important to think beyond the primary motor cortex, or M1, when we think about motor control. So that's the first thing. Now the second thing is to think about what damage to primary motor cortex, or the associated long tracts, produces. So it produces a predictable pattern of deficits. This is the one we think about: loss of movement, weakness, or paralysis. And that's in contrast to what damage to secondary cortex produces. It's a distinct set of deficits, like deficits in motor planning. And so that's sort of general intro point number 2. General intro point number 3 is these key anatomical features of the long tracts that can be used to link clinical presentation to site of damage. Namely, number 1, information crosses the midlines such that primary cortices relate to the opposite side of the body. And number 2, the cortices are somatotopically organized. And I'll just add to that this somatotopy extends beyond the level of the cortex. So along the pathway of motor information traveling through the nervous system, there is some somatotopic segregation. So just keep those 3 things in mind before we start. Now the spinal cord is the conduit for sensory and motor information traveling between brain and body. And the spinal cord is sort of opposite to the brain in that white matter's on the outside, gray matter's in the middle. The white matter surrounding the spinal cord is divided into 3 main columns: a dorsal column, in fact 2 dorsal columns, 2 lateral columns, sort of shown segregated here, lateral column of the spinal cord, and a ventral column, obviously located ventrally, and I'll trace it in cross-section as well. The central gray matter of the spinal cord, sort of in the shape of a butterfly, is divided into laminae based on cytoarchitecture. So you can think of these are called Rexid's laminae, and you can think of these as Brodmann's areas of the spinal cord. So exactly as Brodmann did for cortex, Rexid divided these up into functional areas based on their cytoarchitecture, or their cellular structure. And like Brodmann's areas, the structure did actually map fairly well to the function. Now we tend to simplify the anatomy of the spinal cord by dividing the gray matter up into motor and sensory areas. And if we look at this schematic representation of a cross-section of the cord, we can see that we've got sensory areas located dorsally, and motor areas located ventrally. And in this diagram, they further subdivided that. So because autonomic neurons tend to be sort of in the intermediate gray matter, we put autonomic visceral sensation, intermediate dorsal, coloring red on red, but hopefully you can see where I mean, and autonomic efferent information, intermediate ventral. Now the schematic is conceptually correct, but it doesn't actually represent an accurate slice at any location along the spinal cord. Here's what's wrong. The schematic suggests that the spinal cord is anatomically similar along its length, and it suggests that all the nuclei governing motor, autonomic, and sensory function are present or equally represented along its length. And both of those are incorrect. So in fact, the spinal cord changes fairly dramatically in structure from rostral to caudal. And you can see this in the schematic. So looking at the cervical cord and seeing how different it looks in appearance compared to the sacral cord. and even the thoracic cord. So dramatic differences at every level. And the way the spinal cord changes is in its relative proportion of gray matter and white matter at different levels, in the shape of the dorsal horn and the ventral horn, and most importantly, in what's contained, the contents of the gray matter and white matter at different rostral caudal positions along the cord. So let's take a look at that. Now, the first important principle is that the gray matter of the spinal cord, within it, motor neurons innervating specific muscles and muscle groups are organized into columns. So we tend to look at the spinal cord like this, in sort of a two-dimensional view, looking at a single transverse slice. And in it, we see groups of alpha motor neurons. And in this case, we're looking at alpha motor neurons that innervate distinct muscles. It'd be easy to think that those alpha motor neurons are only present at that segment, but of course they're not. They're present at multiple sections throughout the spinal cord. So in this experiment, a tracer was injected into two muscles, the medial gastroc and the soleus, and then we can look at the ventral horn neurons, the motor neurons that took up that tracer from the muscle. And we can see that those motor neurons supplying those muscles span multiple segments of the spinal cord. So that's sort of the first important feature. Thinking about the spinal cord as having three-dimensional structure where all of the gray matter and the white matter is organized in columns. Now, the somatomotor columns have fairly predictable rostral caudal distributions in the spinal cord. For example, the arm, and by that I mean the muscles between the shoulder and the elbow, are innervated by motor neurons that are present in roughly C5 to C8, while the forearm, between the elbow and the wrist, is innervated by motor neurons present in C6 to C8, just a bit of a caudal shift in the position of that cell column. The erector spinae, conversely, is innervated by neurons in this named ventromedial cell column that are present in all segments of the spinal cord. So in addition to certain cell columns being present only rostrally or caudally at specific distributions or territories, there's some mediolateral somatotopy to organization of the ventral horn. Now, in this schematic, we can see somatotopic segregation that occurs in the ventral horn in two ways: of motor neurons innervating proximal and distal musculature, and of motor neurons innervating flexors and extensors. And the upper limb that's superimposed on the ventral horn over here serves as a mnemonic to remind you how the somatotopy generally works in the ventral horn. So in general, motor neurons innervating flexors are located dorsal to motor neurons innervating extensors, and motor neurons innervating proximal musculature are located medial to motor neurons innervating distal musculature. So let's look at that for a few groups. If we take some distal muscles, like the flexors of the hand or the foot, we can see that those motor neurons are located relatively laterally in the dorsal horn, and the flexors, motor neurons innervating the flexors, are located dorsal to motor neurons innervating the extensors. And we can repeat that pattern moving from distal musculature to proximal, medially through the ventral horn. And the flexor extensor pattern breaks down a tiny bit for the most medial musculature, the muscles innervating the trunk. But this is the general pattern for most of the body. Now, the really important thing to remember is that this diagram only works in combination with that chart. So none of these, you would never see a spinal cord segment where all of these motor pools are present, right? This is a schematic illustrating the somatotopy, but it only works in combination with this chart because you wouldn't ever have the flexors of the hand and foot in the same spinal cord section. So now let's look at the spinal cord along its length and see how it changes, and we'll begin at the sacral spinal cord. So we'll begin right at the bottom. So if we start here, and these are, sometimes I find it easier to look at sort of a holistic picture. So I like this schematic, which contains spinal cord segments from every portion of the cord. So we'll start down here. This is the sacral spinal cord. Of course, these actual size things don't work because this figure's been sized and resized. But if you think about how big the cervical spinal cord actually is, it's about the diameter of your baby finger. So it is small. The sacral cord is even smaller. So it's the smallest bit of cord in total diameter. It has, if you look at it in section, it has a lot of gray matter surrounded by a relatively thin rim of white matter. Now this makes sense if you think about what's in there. So the sacral cord houses parasympathetic neurons, as well as some motor neurons governing the lower limbs. So this creates a substantial gray matter volume. But there's very little white matter because most of the cortical spinal tract and other white matter tracts that are descending have terminated by this time, and very little sensory information has entered the cord by this time. So there's a really thin rim of white matter in the sacral cord. As an aside, although we're focusing on motor control here, if we look at the dorsal columns, only the fasciculus gracilis is present because no sensory information has entered from the upper limbs. We're only looking at some of the sensory information from the lower limbs. Now, if we move up to lumbar cord, here's an L5 section and a section from L2, we can see that the spinal cord is slightly larger in total diameter. It still has a substantial gray matter volume, and it has more white matter relative to the sacral cord. Now, again, this makes sense. The lumbar cord houses motor and sensory neurons innervating the lower limbs. And at the enlargement, which is L4-5, you can see how big the ventral horns and the dorsal horns are, for that matter, with motor and sensory information of the lower limbs. They're a little smaller in L2, where we moved rostral to the lumbar enlargement. Now, the volume of white matter relative to gray matter increases as we move rostrally, because now it contains information associated with additional spinal nerves. As we move up the cord, you can think of spinal nerves being added and added and added progressively. That increases the amount of white matter. If we go to the thoracic cord, here's a section of T10 in the top right, T5 bottom left, we can see that it's small in diameter, relatively small, certainly compared to the cervical cord that it's next to, with scant gray matter and relatively abundant white matter. So if you compare the thoracic cord and the sacral cord, they're almost inverted from each other in terms of relative proportion of white and gray matter. And this makes sense because the thoracic cord isn't containing large chunks of motor neurons innervating limbs. So the ventral horns here mostly contain motor neurons innervating the proximal musculature of the trunk. There's also some autonomic neurons, autonomic efferents, that are present, and they're present in this little IL, the intermediate lateral cell column, that pokes out in the intermediate gray matter of the thoracic cord. The cervical spinal cord is the largest in diameter, still small, but large in diameter relative to the other sections of the cord. It has abundant gray matter, and particularly at the enlargement, which houses motor neurons innervating the upper limbs, as well as abundant white matter. So if you look at the cervical cord in section, here we've got 3 sections, C3, C5, and C8. You can see that they're large oval sections and that both white and gray matter are relatively abundant. The spinal cord receives its blood supply segmentally by branches from major arteries. And the anatomy and details of this is somewhat complex, but essentially each segment of spinal cord is supplied by 3 named arteries that run longitudinally along its length. So we have a single anterior spinal artery, the ASA, that's shown here on this diagram in purple, and it supplies approximately the anterior 2/3 of the spinal cord. And then we have a pair of posterior spinal arteries shown in green, which each supply about sort of 1/2 of the posterior third of the spinal cord. Now, what you can see in this diagram, it shows it nicely, is that there's quite a bit of overlap in the territory of these arteries within the spinal cord. and they're connected and reinforced by many additional arteries. Now, partially due to this redundancy, spinal cord strokes are relatively rare events compared to strokes affecting the brain. So spinal strokes comprise about less than 1% of all strokes. When they do occur, they're most commonly due to occlusion of the anterior spinal artery. So this slide gives you an idea of what territory of the spinal cord could or would be affected by anterior spinal artery stroke. And in order to think about what would happen if you lost blood flow to a single posterior spinal artery, you would have to divide this territory roughly in half. So this would be the territory for that artery. So these are relatively rare clinical events. They do happen, but of course, they're interesting ways to think about functional neuroanatomy of the spinal cord. So now let's talk about the flow of information through the major lateral and medial motor systems. So motor control is governed by interactive and hierarchical feedback loops. And we've already said that sensory input is crucial, but for the sake of simplicity, it's been eliminated in this little schematic. So we know that the basal nuclei or basal ganglia both provide crucial feedback to primary motor cortex via the thalamus. So these two systems have indirect influence on motor control. They are not themselves connected or projecting to lower motor neurons in the spinal cord. Most of the outputs to those ventral horn neurons and interneurons arise from the cerebral cortex and from the brainstem. And we can categorize these outputs based on their location of the location in the white matter of the spinal cord. So we categorize them as being part of the lateral motor systems or medial motor systems. Now, lateral motor systems, as the name suggests, travel in the lateral columns of the spinal cord and synapse on more lateral groups of motor neurons and interneurons. And due to the somatotopy of the ventral horn, we know that this means that they control more distal musculature. In contrast, medial motor systems travel here in the medial portion of the ventral columns of the spinal cord, and they synapse on more medial motor neurons and interneurons governing proximal musculature. Now, this table summarizes the lateral and medial descending motor systems, and we're going to talk about a few of them in detail. We'll talk about the lateral corticospinal tract, of course. We'll talk about the anterior corticospinal tract and the vestibulospinal tracts. And the reason we're skipping some is that some other systems have limited or uncertain contributions to human movement. One example is the rubrospinal tract, which descends from the red nucleus in the midbrain, had to think about that, and mostly terminates in the cervical spinal cord. And for decades, people have been debating the relative contribution of the rubrospinal tract to human movement. Another thing I want you to note is that most lateral corticospinal tract axons synapse on interneuronal pools in the spinal gray matter, not monosynaptically on motor neurons. So they influence motor control indirectly. We're always shown a schematic of a corticospinal tract axon descending to the ventral horn and synapsing directly on a lower motor neuron. That is not the case for control of most muscles. The notable exception to that is the intrinsic hand muscles, mediating dexterous fine movements in the hands. Those are monosynaptically directly innervated by lateral corticospinal tract axons. The third thing I want you to note is that many or most muscles innervated by medial motor systems are actually bilaterally innervated. And so that's really important for postural control and trunk control. Many of those medial motor systems bilaterally govern that proximal musculature. So let's start with the corticospinal tract. So the corticospinal tract arises from multiple cortical areas. We all think of the CST as arising solely from primary motor cortex, shown here in red, Brodmann's area 4. In fact, there's also substantial corticospinal axons arising from premotor cortex and supplementary motor area, primary somatosensory cortex, and even somatosensory association cortex. And so all of these areas contribute to the corticospinal tract. There's actually relatively recent data sort of debating and hypothesizing about the role of these somatosensory neurons that descend as part of corticospinal tract. And they're thought to modulate sensory input as part of motor function. These axons coalesce and condense into the internal capsule and descend toward the pyramidal motor decussation. Now, at the motor decussation, the medulla, about 85%, I often read 90%, of the axons cross to form the lateral, that's an L, lateral corticospinal tract. The remainder descend uncrossed as the anterior corticospinal tract. And so the lateral corticospinal tract is, of course, the main lateral motor system. The anterior CST, the relatively small anterior CST, is a portion of the medial motor systems in the spinal cord. Now, when you look at this slide here, you see corticospinal and cortical bulbar tracts. So this is a reminder that with the corticospinal tract axons, the cortical bulbar tract axons arising from the face and head regions of motor cortex and other cortices travel with the CST. So in this schematic, those cortical bulbar axons traveling cortex to brainstem bulb, are shown in red. And alongside the corticospinal axons, they coalesce in the internal capsule and they exit at different levels of the brainstem. And they're innervating cranial nerve nuclei in the brainstem. So that's what the cortical bulbar system is doing. Now, there's some segregation of the cortical bulbar tract governing muscles of the head and face, and the corticospinal tract governing musculature of the body at the level of the internal capsule. So if we envision these axons, this is all white matter, and they're sweeping from the corona radiata up here, coalescing in a funnel into the internal capsule as they descend. What that internal capsule looks like in transverse section is this characteristic white matter V here. And so that white V you can see bilaterally is squishing axons in amongst the deep brain nuclei. And so it's sandwiched by the lentiform nucleus laterally. I always remember lentiform lateral to internal capsule. By the thalamus at the back medially, and by the caudate at the front medially. So caudate, thalamus, lentiform, and between them is the white matter of the internal capsule. If we look over here, we can see the named regions of the internal capsule. So there's an anterior limb, and of course that's between the caudate and the lentiform. The posterior limb, which is between the thalamus and the lentiform, and an intermediate zone, the bend, called the genu, like knee. So, depending on the source you read, the cortical bulbar tract is located either in the genu or in the most anterior portion of the posterior limb, while the corticospinal tract is organized somatotopically within the posterior limb, arm, trunk, leg. Now, as those axons descend, the internal capsule continues into the cerebral peduncles, or the crus cerebri, translated literally as the feet of the brain. So if you look at the brain from an anterior view, here's obviously the brain stem, we see the pons and the medulla, and the cerebral peduncles, or the crus cerebri, are these stalks of brain right here. And they connect the cerebrum to the brain stem. If we look over here in coronal section, coronal, we can see this is the corona radiata up here. This is the internal capsule squished down between the brain nuclei, here and here. And here is the crus cerebri, bilaterally. So that's where those axons enter the brainstem. And if you take a transverse section here, this is the level of the midbrain. So the cerebral peduncles are visible in a midbrain section. And this is that midbrain section that you see right here. So again, a transverse plane taken right about here in the midbrain. And most ventrally are those crus cerebri. And of course, we know they're located ventrally because we can see them in an anterior view of the brain. And so in the ventralmost portion of the midbrain, we have the cerebral peduncles. That ventral portion is called the basis pedunculi. And about the middle third of those contain the cortical bulbar tract and cortical spinal tract fibers, again arranged somewhat somatotopically, medial to lateral. Now from the basis pedunculi of the midbrain, the corticospinal tract continues through the ventral pons. And the ventral pons, it actually has to travel amongst a whole bunch of scattered nuclei. And then at the most ventral part, it coalesces as the most ventral part of the medulla and crosses the midline at the cervicomedullary junction, or the pyramidal decussation. Now, the reason this is an anatomical structure, the medullary pyramids, and this gives rise to the name the pyramidal tract, although this is somewhat ambiguous since the medullary pyramids also contain other axons, like the rubrospinal axons. But this is why the lateral corticospinal tract specifically is often referred to as the pyramidal tract, while other motor systems are often described as extra-pyramidal tracts. Now this is interesting. It's another area of neuroanatomy where there's been a substantial recent update. So as axons travel in the lateral CST, the lateral corticospinal tract, within the lateral funiculus of the spinal cord, it was previously thought that there was clear somatotopy, such that axons governing the upper limbs were medial relative to axons governing the lower limbs. So medial to lateral somatotopy within the lateral CST. Now this model has been updated. The lateral CST is now thought to be composed of all kinds of non-segregated dispersed fibers. So all of the axons within the CST innervate the hands, the arms, the thorax, and the legs. However, the reason the H's are big in this model is that the corticospinal tract itself plays an outsized role in hand function specifically. So it's not somatotopy within that tract, it's actually the role of the corticospinal tract governing hand function that gave rise to this idea of somatotopy. Now the remaining 10 to 15% of corticospinal tract axons continue beyond the medulla without crossing and enter the anterior white matter columns of the spinal cord to form the anterior CST. And just as a reminder, this is what the schematic's showing, so I've made it a little bigger for you. Many of these axons bilaterally innervate proximal musculature. So there's bilateral innervation of a lot of this trunk musculature from these medial motor systems. Now, in addition to the anterior corticospinal tract, 3 other medial motor systems or tracts travel in the medial ventral column of the spinal cord. These originate in the brainstem and terminate on interneurons or motor neurons in the ventral horn, and they're sometimes referred to as having indirect influences on movement. So of these, we'll talk just a little bit about the vestibulospinal tract. And the vestibulospinal tract is actually 2 tracts, and confusingly, they are named lateral and medial, even though both are part of the medial motor systems category. They just happen to be lateral and medial relative to each other. So these nuclei originate in the pons and the medulla. Nuclei for these tracts, I wrote medullar in the medulla. There we go. In vestibular nuclei that receive input from, you guessed it, vestibular organs. So it's vestibular organs in the inner ear that send input to these brainstem nuclei. And they in turn, originating in the pons and in the rostral medulla, descend as the medial and lateral vestibulospinal tracts. Now they have slightly different roles. So the medial vestibulospinal tract primarily reaches the cervical and upper thoracic cord. It's important for stabilizing head muscles and also important for coordination of the head and eye muscles. And you want your eyes to move when your head moves. The lateral vestibulospinal tract, in contrast, projects to all levels of spinal cord, and it activates primarily extensor motor neurons, so opposing flexor tone. Both of these are important for balance and posture. So damage to the vestibulospinal tract results in vertigo and ataxia, loss of coordination. Now we'll end this little session by talking about clinical descriptions of motor deficits, and we'll move to this as a tutorial activity. Now motor neurons are described clinically in three main ways: as upper motor neuron lesions or lower motor neuron lesions, first distinction, by the severity or nature of motor dysfunction, and by the location of motor dysfunction in the body. So of course we know there are upper motor neurons and lower motor neurons. And this is true for both control of the body and control of muscles in the head and face. So in the case of the body, those upper motor neurons in the cortex travel from the brainstem to the spinal cord in the corticospinal tract and impinge on lower motor neurons or their associated interneurons in the ventral horn. In the case of cranial nerves governing the head and face, those originate cortically, descend as the cortical bulbar tract, and innervate lower motor neurons that are mostly located in the brainstem, the cranial nerve nuclei. Well, both types of injuries to the upper motor neuron or its axon, or the lower motor neuron or its axon, produce weakness or paralysis, the nature of the clinical presentation is somewhat different. So upper motor neuron lesions produce weakness. They produce less atrophy compared to lower motor neuron lesions. So there could be some atrophy due to disuse, but there's not a complete lack of tone. In fact, reflexes and tone tend to be increased in upper motor neuron lesions, at least after the acute period. And this is thought to be due to loss of descending inhibition of pathways nearby the corticospinal tract. One of the things that comes back in some upper motor neuron type lesions is the pathological Babinski reflex. So the Babinski reflex, remember, is normal toes. If you stroke the bottom of the foot, the toes normally curl in flexion in a normal adult. A positive Babinski reflex in an adult with an upper motor neuron lesion, the toes will splay out and extend like they do in a baby. Clinical language also describes motor deficits in terms of both their severity and their location. So plegia and paralysis indicate no movement, and that means no voluntary movement. You can have reflexive movement, but no intentional or volitional movement. Whereas paresis indicates partial weakness, and you can describe that as mild weakness, moderate weakness, or severe weakness, depending on the score you would get on a neuro exam. Palsy is interesting. It's an imprecise term for weakness or no movement. So it's a bit of a less precise clinical term. You can denote location with hemi, which is one side of the body on the mid-sagittal plane. Paralysis, for example, or paraparesis, is weakness of both legs. Monoparesis is one, weakness of a single limb. And di is both sides of the body are equally affected. So facial diplegia, symmetrical facial weakness, for example. Finally, quadriplegia or tetraplegia is paralysis of all four limbs, quad or tetra.